you walk us through the study in the Australian senior citizens that I think for many people was I don't know, for me at least, a huge turning point in how we thought about this drug.
And all I'll say to me is that that paper, the Joan Mannick paper with rejuvenation of the immune system, I think will be seen in the aging field as certainly a milestone paper along with the ITP paper, because as far as I know, it's really the first where you actually rejuvenate some organ system in a human being, right? And so I think her study really was mind-blowing. I think Matt can speak more to the details of it. And,
I would agree completely with that. And we may want to come back and touch on that, because I think as people are thinking about clinical trial endpoints for Gerotherapeutics, that's a perfect case example of a functional endpoint that you can actually do a clinical trial on for FDA approval and show improvement in function and potentially get a drug approved as a gerotherapeutic. So I think as a conceptual advance, it's important as well. So just to give a little bit of history, there was actually a paper, I think it was 2009 from Pen Zheng's lab in mice that preceded the Joan Mannick paper, where they showed that you could treat with rapamycin for, I think, 6 weeks in that study, and rejuvenate the immune function of a mouse. And to me, the one experiment in there that is most compelling is they have a set of mice. I think they were 24 months of age when they started this experiment, and then they had young mice, and the mice got either a flu vaccine or no vaccine. And then they waited, and then they gave them what would be a lethal dose of influenza if they hadn't been vaccinated. And then in the aged mice, they either got rapamycin for 6 weeks or they didn't. And so if you're a young mouse and you don't get a vaccine and you get this dose of influenza, there's 100% mortality within, I think it was 8 days. That makes sense, right? No vaccine, you're not protected against the influenza. If you're a young mouse that got the vaccine, 100% protection. So that, again, makes sense. It's a control. If you're an old mouse, no rapamycin, you get a vaccine, only 30% of the mice actually were protected. So this is showing you the impact of just normal biological aging on the ability to respond to a vaccine. In mice, it's about 70% of the time you don't respond to the vaccine and you die then if you get a subsequent influenza infection. Interesting parallels to humans, as we've learned over the last 4 or 5 years. The cool thing in that study was if the mice got 6 weeks of rapamycin treatment before the vaccine, they were then 100% protected. These are old mice. They're now almost 28 months old. So this is sort of an amazing demonstration of immune rejuvenation in an aged mammal. So I think that study is what really set the stage and allowed Joan and the group from Novartis to be able to move forward and convince the people who had to fund this study that there was a reason to think that mTOR inhibitors might do the same thing in humans. So the design of that human study conceptually is very similar to the mouse study I just laid out, except, of course, they didn't give people lethal doses of influenza. But what they did do was they enrolled healthy older people. I think they were over the age of 65, and they couldn't have, there was some set of pre-existing disease that they would be excluded for. So they were considered relatively healthy for their age. And they got either placebo or I think in the first study, they tested 3 different doses of everolimus. So it wasn't rapamycin, but I think we can just think about it the same as we would rapamycin based on our earlier discussion. So there are a few interesting things here. So they got, I think, everolimus for 6 weeks or a placebo, and they got either
I think it
5
was,
milligrams.
Yeah, it was 5 milligrams once a week, 20 milligrams once a week, and I think it was 1 milligram daily was the third
That
group,
was what I was going to say.
placebo.
So I think between the two of us, we're close, if not spot on. Yeah. So there are some interesting, and then they gave, so that was for 6 weeks, and then they gave a flu vaccine. And then they looked at antibody titers and I think, I don't know if it was this study or a later one where they looked at viral gene expression as well. And then also subsequent infections over the next, 6 or 12 months or something like that, respiratory tract infections. So what the first paper showed was, I think, pretty convincing data that at least at the 5 milligrams once a week and 1 milligram daily dose, there was a boost in response to the vaccine as measured by antibody titers. So that supported the idea that, similar to what had been shown in mice, you could, in fact, to some extent, rejuvenate the ability of the aged immune system in humans to respond to a vaccine with transient dosing with a rapamycin derivative, everolimus in this case. The other thing, though, that I think was super important about that paper was it was pretty large. I mean, not huge. It wasn't like a phase 3, but there were
320 if I'm not. Yeah, I think it was about 80 per arm.
Okay, so hundreds of people in this study who got everolimus who didn't have an organ transplant and weren't taking other immunosuppressants. And the side effect profile, at least in the 5 mg once a week group, was essentially no different than placebo. And so I think that study started, and it's been slow because there's still a perception that rapamycin has a lot of bad side effects, but that started at least some people in the community thinking, maybe, and this I think is getting what you were talking about, Peter, maybe it is possible that lower doses of a rapalog in relatively healthy older adults could be well tolerated. And maybe this idea that as a gerotherapeutic, we might be able to give rapamycin to older people, maybe it's not so crazy. I think that's one of the important aspects of the study, kind of independent of the potential immune rejuvenating effects, which I don't want to minimize because that's hugely important. I actually think both of these things are important things that study kind of set the stage for.
And I think from that study comes a word, I don't know if it was certainly wasn't coined in that study, but in my mind, at least, it went from, we shouldn't think of this as an immune suppressant. We should think of it as an immune modulator. Right. And that was a clear example of how you take at least an aged immune system and make it more robust.